Got it. All right. Hold on. There it is. All right. We had exam on Friday. You should get that back no later than Friday. All right. So about halfway done graded. Hope to finish grading the rest of it today. So if we get it done today, then we'll get it back on Wednesday. If not, it'll Friday. Here in class. We have a quiz that's posted. I think I said that due date was Tuesday, maybe? Tuesday night? Get you a chance to get back, review the notes today, tomorrow, kind of get back into this physiological ecology section, especially with water balance. Lab is biodiversity. So bring those sheets, that Excel document. We're going to be using it. We're going to use the calculations again. It's all in lab, just a lot of calculations. And then next week we are outside. We're going to drive to the red arroyo. We're going to mark recapture grasshoppers. So if you can drive and if you can take people, that would be great. Next week we will meet down in the lab, take our quiz. They'll get a brief description of what we're doing and where we're going, and then we'll meet out there. That lab we mark for like an hour, and then once we're there, I'll collect the nets and everything else and bring them back to the lab. So if you wanted to leave from there, you can. Just remember, if you brought people, if you took people there, make sure you can get them back. All right. So it's rain or shine. So if it's rainy out, drizzly out, we're still going out there. Plan for it. And there could be mosquitoes. There's been a few days where it was relatively still, limited wind, and mosquitoes were out. But for the most part, when we're out there, for the vast majority of the days when we've done this, the wind is usually blowing, and it might be hot, but the mosquitoes didn't seem to bother us too much. The things that are out there are all of those burrs. There's quite a few burrs out there, so might want to wear pants, wear a hat, sunscreen if you're sensitive, and so forth. All right. So this is where we left off. We were talking about water balance in plants. We talked about the problem of drying out, right? Increased stress. You have possibility of having the entire water transport system fail, and carbon starvation. And the water really comes down to the water in the soils, because that's where the plants are taking it up. That's a primary site of absorption. So the amount of water in the soil is the difference between these two terms. Field capacity is how much water can actually fit between the soil particles. That permanent wilting point is how much water gets stuck to those particles and thus can't move into the plant roots. But that water, the amount of water that's there, is only part of the story. All right, we have to consider the salts in those soils, because the plants are going to set up a situation where the water is going to flow down its gradient. So the more salts there are in the soil, saltier the soil, the saltier the plant is going to have to make its root hairs to try to promote water to flow into them, and that costs energy. Now, do plants actually do this? Yeah, plants do this. We have great examples of plants that can survive salty soils and have the adaptations for these salty soils. So we know it's out there. It's a problem that the plant has to address. All right, so that's where we left off. All right, a lot of times when we look around at various ecosystems around the world, we see these hot areas tend to also be dry. They're going to be dry. So how do the plants evolve? What are the mechanisms? Well, we have to first understand the problem, and the problem is related to drying out. All right, so the plants have stomata. They're going to open for gas exchange, but if they open, you're going to have an increased amount of evaporation. So the plants are going to try to control how much water they lose by closing the stomata. But if the stomata are closed, then you can't get gas exchange and you can't proceed with your photosynthesis. So, what kind of adaptations have evolved to allow our plants to become successful in these hot and dry conditions? The solution comes down to where we're going to fix the carbon and assimilate it into organic molecules. All right, so we've got carbon fixation and assimilation. We've got two different ideas. We're gonna define what those are. When we talk about carbon fixation, we're taking CO2 out of the air and getting it into an organic compound. All right. The assimilation part is where the CO2 in that compound now gets integrated into our metabolic cycle to produce our sugars. All right. In some cases, the fixation and assimilation happens in one step. In other cases, we'll have a fixation step, and then we'll have an assimilation step. So there's two different processes. And our adaptations to survive these hot and dry conditions involve changing where and when fixation and assimilation is going to occur. All right? All right. We got some static. Hold on. All right. Let's hear about the static. Is the static still there? Where are my settings? Oh, I would mute them, maybe. Oh, okay. Yeah, just let me know if you get the static. I just unplugged the microphone and plugged it back in. Seems to work. All right. So we're going to separate fixation and assimilation. Let's talk about how they do this. We're going to start with the C3 cycle. This is your Calvin cycle. All right. This is the original way that plants did this. It's probably evolved first. All right. So here, our fixation and assimilation occurs in a single step. We're going to take our CO2 out of the air, and when we get it into that organic molecule, the organic molecule is already in our metabolic pathway, our metabolic cycle. All right, so how does it get fixed? It's going to get fixed and assimilated into 2, 3 carbon molecules, which means we're going to start with a 5 carbon molecule. When we add the carbon from the CO2, that makes 6, but it's actually going to produce 2 of these 3 carbon molecules. The reaction itself is going to be catalyzed by this molecule here, this Rubisco. Rubisco is ribulose bisphosphate carboxylase. Carboxylase. How did I misspell that one? Carboxylase. That's better. All right. Ribulose bisphosphate carboxylase. It's going to capture the CO2, combine it with ribulose bisphosphate to produce a 3 carbon molecule, 3 phosphoglycerate. And it's going to produce 2 of them because we need to have, if we start with 5, this is our 5 carbon molecule, and we add 1 more, we're going to get a total of 6. So we get 2 of these out. All right. This Rubisco is found in the chloroplast. So the CO2 diffuses in through the stomata into the chloroplast where Rubisco is going to be found. And then Rubisco captures it, gets it into this 3-phosphoglycerate, which then cycles us all the way down to get this G3P molecule, this other sugar molecule that goes out. All right, so once we get here, we're going to pass the energy on to the energy production steps, and that's where we get our sugars and so forth. But once we get to this step, this G3P, we're going to have to regenerate the ribulose bisphosphate. All right, because it's a cycle. So nothing really gets consumed. We're just capturing the CO2 and transferring it down to here. All right, this takes energy. It's going to take energy to regenerate our ribulose bisphosphate. And that energy that it needs is going to come from the light reactions of photosynthesis. All right, so your photosynthesis has your light reactions, the photoreceptors, or photosystems 1 and 2. That's going to produce the ATP that is needed for this cycle. And then all the extra energy gets produced from here once we get to the outward. All right. Now, I think cell bio covers this, maybe botany too, but I know you could go into even more detail over these steps. We don't need to do that because what we're focused on are the adaptations that have allowed our plants to survive. So for this step, we need to know a couple things. One, we have a single fixation and assimilation step, and that step is catalyzed by Rubisco. All right, and then the energy needed to regenerate your RUBP comes from our light reactions. Those are important concepts. All right, so Rubisco is going to capture the CO2, but its affinity is only considered moderate affinity. All right, so the affinity is how well will it bind to the CO2 when it's there. It only shows a moderate affinity for CO2, and that has consequences. Because if you have something else that Rubisco can bind to, then Rubisco might bind to that compound and thus stop working to fix and assimilate the CO2. All right? So we have a term for that concentration of CO2 needed to ensure that we are going through this cycle, and that is called the Co2 compensation point. The CO2 compensation point. This is a CO2 concentration at which point photosynthesis balances respiration. So let's look at a graph. I don't know if you're aware, I grabbed a new thing in chalk. No more these tiny pieces. All right. Our axes. X-axis is our carbon dioxide concentration. This is the concentration right at Rubisco. All right, it's right at that enzyme. Why did I say right at that enzyme? Because if the stomata closed, your concentration outside that leaf is going to be different from the concentration inside. So we're focused on the concentration right at that enzyme. O2 generation is our y-axis. All right. So if photosynthesis is proceeding, what gets produced? Oxygen. All right. So if we are binding, fixing, and assimilating, we should show net generation of oxygen. So that is going to mark our 0 point. So we have net production, this photosynthesis occurring. Photosynthesis occurs because we're in the positive. Down here, we're in our negative. Now our photosynthesis is not occurring. And we'll have a name for that. So what does our graph look like? Well, for a C3 graph, we're looking something like this. All right, CO2 concentration at that enzyme increases in concentration. We're not producing any oxygen until we get to this point. This point here, that is our CO2 compensation point. This is the point where our photosynthesis completely balances respiration. Because if we're not producing energy, we're still consuming energy. All right, we're still consuming energy. That's our break-even point. So for C3 plants, we have to have this amount or more to photosynthesize. What happens when we drop below it? Well, Rubisco has this interesting property in that it doesn't just bind CO2, it also binds oxygen in a process called photorespiration. So in this case, here for the C3 plants, we have photorespiration occurring. Get that autograph. For this, we have photorespiration occurring. Oh, oxygen is binding in a different pathway. So here's our graph. Shrink this up. Here's our example pathway. You have CO2 that typically binds Rubisco. It produces your 2-PGA. All right, it gets dumped into our pathway, and then it regenerates ribulose bisphosphate. All right, that's our typical pathway. That's our fixation and assimilation. If we have photorespiration, oxygen binds to the RUBP, but it produces these two molecules. All right, one of them we use to regenerate RUBP. The other one we don't. We don't regenerate. So we basically take this 5-carbon molecule, split it off into a 2-carbon and a 3-carbon molecule, but then we have to have another 3-carbon molecule around to regenerate this RUBP. And that costs even more energy. So this process here already costs energy to regenerate RUBP. Once we start binding oxygen, it's consuming even more. The end result is what we get, that carbon starvation. You get photorespiration. We're no longer producing energy. Instead, we are consuming energy, and we're consuming energy at a faster rate now than what the plant would typically consume. So if we close down our stomata, we might close it down and we might have this amount of CO2 that's in our plant. But as we are doing photosynthesis, we are gradually reducing our concentration. And we hope that the plant can open those stomata before it breaks through this compensation point. If it does, great, we're back up high. We're still photosynthesizing. But if those stomata stay closed for too long, we're going to drift down here, oxygen's going to start binding Rubisco, and then we're going to rapidly consume our energy and potentially kill off the plant. It's not very good for a dry environment, for a hot and dry environment, right? So what kind of adaptations do we have? First adaptation is called the C4 pathway. So the C3 pathway, I'm going to go back to this graph, was named because when it captured that CO2 from the air, it made a 3-carbon sugar molecule. It made 2 of them, but it made a 3-carbon sugar molecule, 3-phosphoglycerine. So hence the C3. In a C4 pathway, the plant is going to capture that CO2 and get it into a 4-carbon sugar molecule. All right. Now, this is a separate pathway because what the plant is now going to do is to separate our fixation step from the assimilation step. And the C4 plants do it by separating it in space. So we're going to do fixation in one location, we're going to do assimilation in a second location. All right, and our assimilation is still the carbon cycle, or the Calvin cycle. Same cycle that appears in the C3, but our C4 plants have an additional step, an additional pathway. So what is that pathway? That pathway is to take CO2 and fix it into a 4-carbon molecule using the enzyme Pep Carboxylase. So phospholynyl pyruvate, PEP, is going to take the CO2, it's going to be bound to it, and that reaction is catalyzed by phospholynyl pyruvate carboxylase, PEP-C. All right, so we're going to get PEP plus CO2. This step gets catalyzed by Pep-C to produce oxaloacetate, OA. And again, OA will work. And then that OA gets transformed into malate. Malate, for us, is going to work as our transport surface. So malate will then move, that malate now has our captured CO2. Malate's going to move that CO2 into the chloroplasts where Rubisco can be found. All right. And then the malate's going to dump that CO2 to Rubisco to basically ensure that wherever that Rubisco is, we're always going to see a higher concentration of CO2, so we can always proceed with photosynthesis. So this is Rubisco. This is C3 plants. What about C4 plants? Well, C4 plants, this whole process is governed by that Pep Carboxylase, Pep-C. And Pep-C has a very high affinity for CO2, which means it's going to be able to bind CO2 at a much lower concentration than Rubisco. So if we were to add our C4 plants to this graph, we'll get a curve that looks something like this. Do chalk. That's that. C3 plants have Rubisco. C4 plants are dealing with Pep-C, Pep Carboxylase. So our compensation point, we still have a compensation point, but it's down here at a much lower concentration than our Rubisco. Now, I didn't mess this up. This line is ultimately going to be lower than our C3 plants. We've got trade-offs, right? We always talk about adaptations having trade-offs. This is a trade-off. We do well by having a higher affinity. We don't bind, Pep-C doesn't bind oxygen. So we're not susceptible to photorespiration. But the cost is, if we have high CO2 levels already, we're going to do less well than those C3 plants. And the question is, why? Why do we do less well? Why don't we do as good as those C3 plants? Well, it's because we added that extra step. We added that Pep Carboxylase step where we first fix the CO2 and then get it to malate so we can then shuttle. All right? So with our reaction, we have to regenerate phosphoenolpyruvate. So we get our PEP, it gets to a oxaloacetate, gets to malate, malate shuttles it. Well, once malate releases it, it has to be transported back and then regenerate phosphoenolpyruvate so that we can capture even more CO2. This costs energy. But we also have the Calvin cycle that's also costing energy. So overall, we now have two cycles that are costing us energy. So the C4 plants are going to ultimately be less efficient than the C3, but they do well when we close our stomata because the Pep C can continue to operate as the CO2 concentrations in the leaves start to be consumed. Now, how does this work? Why did it develop? Well, it was developed and it was made even more possible by something called the Krantz Anatomy. And it's the anatomy of the leaf, the anatomy of our leaf structure. So what is this Krantz Anatomy? In the Krantz Anatomy, our chlorophyll and the chloroplasts themselves are concentrated in bundle sheet cells. So these are just two different diagrams. This diagram up here is a C3 cell. All of the green dots, those are the chloroplasts. That's where our light reactions are going to take place. That's where the Calvin cycle is taking place. All right. In a C3 plant, they're scattered all throughout the leaf. So the stomata open, gas diffuses into here. You can have this the Calvin cycle occur all throughout the leaf. That's different from the Krantz Anatomy. Now, notice that there are no green dots here because all of the green is concentrated in the bundle sheet cells. These are these bundle cells, these cells that kind of encircle the vascular system. All right, so CO2 gets into the leaf, but CO2 can't get to this. So what's in all of these cells? That's your Pep Carboxylase. This is where your Pep C is going to fix the CO2. So you have your, let's say you open up the stomata, you got CO2 levels up here, all throughout the leaf, stomata closed. Pep C is consuming the CO2, or fixing it, I should say, into oxaloacetate and malate, and the levels continue dropping. In a C3 plant, once we get to this point, we're at a bad spot. But for our C4 plants, we can keep going. Pep C continues working, and it can continue working during the hottest parts of the day, which is when the sun is shining. So they can still photosynthesize. They can still use the energy from the light reactions to regenerate all that PEP. All right? The malate then is shuttling this CO2 into these bundle sheets to maintain our concentration of Rubisco at the high levels. So we never really run into a problem of photorespiration, of that Rubisco capturing the oxygen, because malate is just constantly dumping the CO2 right where it needs to be to ensure a high concentration of the CO2. All right, so I asked the benefit. I just said the benefit. We can avoid photorespiration. We are ensuring that Rubisco always sees a lot of CO2 and can bind to it. All right, so when the stomata close, what happens in a C3 plant? CO2 gets consumed until photorespiration begins. And you don't have a very long window. C4 plants, Pep C continues using CO2 until it can't use it anymore. All right, we don't have photorespiration. But Pepsi stops working because there's not enough CO2. You still have malate releasing CO2 to Rubisco. The plant can still photosynthesize. And this is all done, this process is all done by separating fixation and assimilation in space. Questions? All right. Leave it on? Leave it up a little longer? Yeah, because what we're going to do is we're going to talk about the cost of this. Yeah, it's great benefit. There's always going to be costs. We've already said one. All right, ready? All right, what are the costs? It's energetically expensive. All right, it's energetically expensive. Not only are we regenerating RUBP in the Calvin cycle, I mean, that's kind of fixed, so that cycle has the same cost as the C3 cycle, C3 plants. But the C4 now have this Pep C cycle. All right, and that has to regenerate Pep, and that's going to consume energy. All right, so we've got extra energy that has to be regenerated there. Also, the transport of malate. Malate's not just going to diffuse easily. We're going to have to use transporter proteins. That's going to cost us energy. So the end result in these two steps is that when we already have high levels of CO2 and we're not at risk of like drying out, C4 plants are going to be less efficient. So if you pit these two against each other at a time where we're not really water stressed, we've got plenty of CO2 around, C3 plants are always going to win. But we know that we don't have these conditions all over the place all the time. We have those conditions where we have that need to conserve water, so we're going to produce situations where we're probably down here at our average CO2 levels, and now we give the benefit to the C4 plants. And that has allowed them to evolve and to persist. Are there a huge number of C4 plants? No, not really. But they're there. They're an adaptation that has allowed survival in these hot, dry environments. Now, I do have this graph on here just because we're going to come back to this. And this is this idea of finite energy. An organism has a finite amount of energy to spend, and it's going to spend it on three different processes. It's going to spend it on maintenance, on growth, and reproduction. So reproduction, hopefully, is obvious. We're going to spend some energy to mate, to reproduce, produce offspring, and in some cases, take care of our offspring. Growth, hopefully, is self-explanatory too. We have to grow, we have to develop. All right, so we have to spend energy on this. This maintenance cost is just your regular basal metabolic rate. If you just sit there, what are your normal costs? This maintenance also includes immune responses for organisms that have an immune system. So if we are increasing our maintenance energy, which is what this regeneration of PEP and Rubisco would be, or RUBP, if we increase our maintenance energy, that means there's less energy to be spread between growth and reproduction. And that's where our efficiencies come into play. We've got less energy available for growth and reproduction. If we're not leaving as many offspring, we're less fit than those that can leave a lot of offspring. So cost number one is that it's energetically expensive. Cost number two is somewhat tied to that. Because the ATP that's needed to regenerate both the RUBP and the PEP is going to come from our light reactions. So that's where we're getting our ATP. We need to be in an area that has a high amount of light. And not just high amount of light for part of the day. We need to ensure a high amount of light so we can continue fixing the CO2 as our concentrations decline. So that means that these plants really can't do well in the shade at all. And if you think about all of the understory plants, if you think about forests and how things evolved and you get plants that grow tall and start casting shade, the C4 plants, they're not going to be very successful in the understory in those spots that have the shade. So it's just kind of like another reason why all of our plants aren't C4. It sounds great, but the efficiency, the inefficiency, and the requirement for a high light condition really hinders the C4 in comparison to the C3. Questions? All right, let's see how far we go. Yeah, 5 minutes. I don't know. I guess we'll continue. We'll continue because then what we could do is review both on Wednesday. All right, so the C4 pathway is one pathway. Oh, so question we had is the C4 adaptation convergent or divergent? Did it evolve multiple times or just once? I believe the C4 pathway may have evolved multiple times. I know this pathway, CAM pathway, I believe also evolved multiple times, but I'd have to look that up. So the CAM pathway. This is a crassylation acid metabolism pathway. We are also separating carbon fixation and assimilation. All right, why? Because again, we want to make sure that Rubisco always sees a high concentration of CO2. The C4 plants separated it in space. These CAM plants are going to separate it in time. So we're going to do fixation during one part of the day. We're going to do assimilation in a different part of the day. And the day, my reference of a day is 24 hours. All right? So fixation in this case is going to occur at night. It's going to occur at night because that's when our stomata are open. All right? Less likely to have a lot of water loss when they're open during the cooler parts of the night. So you're going to have fixation. You're going to capture as much CO2 as you can, and you're going to store it. You're going to store it all throughout the night. And then, once the daylight comes, it's going to heat up. Your stomata are going to close. It's going to pinch off. Now, at that point, your fixation is going to ultimately stop. It's going to still produce. It's going to still work and store. And then it's going to stop. And at that point, now we transition to our assimilation. And we're going to unload all of that stored CO2 right to Rubisco so that we can start our sugar producing steps. Now that's pretty good because during the light reactions, we basically are regenerating the RUBP. And that's good because we're unloading it. We're unloading CO2 there. But we're also using some of those light reactions to regenerate the phosphoenol pyruvate that we need so that when it turns night again, we can start our fixation. All right, the pathways are basically similar. The pathways are basically similar. You have your CO2, It's going to combine with Pep via Pep Carboxylase to produce oxaloacetate. That gets transferred to malate. Malate goes into the vacuole. It's actually malic acid is what's going to be stored. All right, it's going to be stored as malic acid. So that's kind of like a step to, protonate it. And then it's just going to build up in the vacuole, the plant vacuole. When it's time to start assimilating, the malic acid deprotonate allows the malate to come out, and then malate is going to start releasing all of its CO2 right to the Calvin cycle. So here's your Calvin cycle. So in the graphs, you have your malate releases the CO2 to produce your pyruvate, and then you regenerate to phosphoenolpyruvate and so forth. All right. And your Calvin cycle has your regeneration. Now this doesn't have, these plants don't have the Krantz anatomy because we're not separating in space. Instead, we're separating it in time. And this is actually a pretty complex pathway, complex regulatory pathway. Because it's, you have to ask, well, how does the plant know that it's daylight or not daylight? And it's going to be a combination of temperatures and your CO2s, because you're going to close your stomata and so forth. But the temperature is going to be our signal to tell the plant to start producing the enzymes and proteins needed for different reactions, for different pathways. So, when it gets hot, it'll turn on the enzyme production for the Calvin cycle. When it gets cooler, it shuts those off, but allows production of PEP. And there's also inhibitory pathways related to the malates and the phosphoenolpyruvates. So it's all these pathways just increase the capacity or the complexity to ensure that your fixation steps occurs at night, your assimilation step occurs during the day. Because the last thing you want to do is have malate come out, release CO2, and then have PEP bind it right away. You can't have that happen. So all this complex regulatory pathway really increases the costs. All right? So we're regenerating phosphoenolpyruvate. We have the malate transport. All right? That's the same as the C4, but now we have this malate transition to malic acid, and you have all of that regulatory work to make sure that we can separate it between the day and the night. That's all costing energy. So between the CAM and C3, C4, yeah, CAM costs the most. It is the most energetically expensive. When we add it to our graph for our compensation point, you'll see that our compensation point for the CAM is similar to our C4 plants, which is like this. Here's CAM. Basically the same point because we're using phosphoenolpyruvate carboxylase, but notice we barely get above our zero line because these plants consumed a lot of energy, and as a result, they're going to exhibit slow growth rates. You're going to have fewer offspring that get produced. We've just shifted a lot of our energy usage to maintenance at the cost of growth and reproduction. All right, so our C3 or the CAM plants, yeah, less energy efficient, but they are the extreme water savers. You only find them in arid environments. You only find them in high light conditions, like really high light conditions. They do not do well in shade at all because of all of this extra energy that they have to expend just to get the CO2 fixed and assimilated. All right, so look over this. On Wednesday, we'll review C3, C4 CAM, and we'll talk about some of the trade-offs evolutionary trade-offs as to why we see so few C4 and CAMs. Yep. And the attendance is, did I really forget it? 9. 9. That's our answer. 9. The correct answer is 9. And you can, if you checked on Blackboard, I believe I added a column for the extra credit that would be added to the exam based on our attendance. Double check that. I had all the attendance quizzes listed. I think we only had one day where I took attendance. I didn't count the first day. That's just the first day of class. Double check that, because what I'm going to do is start hiding all of the old attendance quizzes. That way we don't see them. I've already logged it. So if you take issue with your attendance, please email and we'll look at it. All right, Wednesday. Some of you have lab. Don't forget tablets, laptops, calculators. All right.